Hello, and welcome to the AK Podcast, where we explore science and nutrition behind livestock care and management with the best and the brightest in the business. I'm your host, Chris Radke, part of the sales department here at Agri-King, and with me today, as usual, is the Director of Field Service and a member of the sales management team, Mr. Mike Donaldson. Mike, how you doing, buddy?
Very well, Chris. 2025's come in pretty good. Our first one of the new year.
Yeah. Do you do any New Year's resolutions, Mike?
No, I stopped fooling myself a long time ago. I just sort of try to do better and don't tie it to a certain date.
There you go. Then you win every time. You
Yes, you write them down, so you do have to hold yourself accountable, but you don't share them with people that going to use it against you.
Ooh. All right, Mike, what are we talking about today, and who's with us?
Well, we've got Dr. Luke Koester and Dr. Adrian Baker from Agri-King. We're going to go over what makes a ruminant, I suppose. Some of the kind of special things about ruminants that make them very different than monogastrics like ourselves. Dr. Baker and Dr. Koester, thank you so much for joining us. Luke, would you like to give a little background, and then we'll go to Adrian for the same, and then we'll get on topic.
I thought this was Adrian's show. I'm just tagging along. Oh, hi, my name's Lucas Koester, and I have my PhD in microbiology, focusing mostly on ruminant microbiology. So both the feed fraction as well as the epimeral communities of that rumen, which are adhered to the wall. So there's a plethora of organisms in there that are super interesting to me, and that just kind of took me on my track as a microbiologist and as an animal scientist, kind of both. So that's where I'm at today.
Perfect. Adrian, tell us a little bit about yourself, please.
Got my PhD in animal science, focusing specifically on more on feedlot nutrition of the beef nutrition game. You've spent a lot of time working on starch utilization, whether it be corn processing or just looking at starch digestibility in feedlot cattle. So that's kind of my background. I would say that was most of the focus of my PhD.
Perfect. Well, just we're obviously Agri-King's a nutrition company. We mostly are dealing with ruminants that are cattle, dairy, or beef. But just to sort of set the field a little bit, what are some of the other animals that are actually classified as ruminants?
Adrian, take it away.
Oh, well, so Dr. Luke and I, we were discussing this the other day, and we kind of, we both, I think, thought back to that, when you get into these grad programs, they always have that, like, first day of class, right? And it's like, well, here's the, the ruminants, and here's what's going on, and here's what are ruminants, and there's ruminants, so that's your cattle, sheep, deer, bison things, and then there's kind of a little subset called pseudo-ruminants, and that falls into camels, llamas, and alpacas, and what makes them pseudo is they don't have an omasum, where ruminants have 4 compartments to their stomach. They have a reticulum, rumen, then an omasum, and then an abomasum.
So when I've spent my whole life saying that cows have 4 stomachs, I've been wrong every time, is what you're telling me.
It's mostly generally assumed it's 4 compartments because it really takes the whole organ, though you can actually technically remove the omasum. That's been done experimentally, and the animals still are able to digest feed. But each compartment has a separate, I guess you'd say, specialization that furthers the digestion for that animal.
Well, let's take maybe walk us through in the order that feed would pass through those 4 compartments. You already named them off, but let's start with the reticulum. What's its purpose? What's it do? Just kind of go A to B to C to D through those areas, please.
Yep. So the reticulum's kind of, if you're thinking about the digestive tract as a highway, right? if you look at a monogastric, things flow down the esophagus, go to the stomach, and then to the small intestine. So ruminants are a touch different because you got the reticulum, and that's, I would kind of, if you're looking at it as like a roadway, I would call it more as a roundabout.
Okay.
Feed enters the reticulum, and from there it has the ability of either going to the omasum. So that's kind of really what most people classify as the third area, but that's not really what happens. It goes to the rumen. Then as part of that, animal ruminates and they're contracting and breaking down those feed particles. Some of that flow will end up back in the reticulum, and then it'll flow to the omasum. And that omasum is kind of almost like a barrier in that roundabout for particle size, essentially. And so it doesn't really go from reticulum to rumen, then omasum. Everything has to go back to the reticulum before it can go on. Is that a fair roundabout?
That's a hilarious and pretty perfect description. So what's the common name for the reticulum, right? Isn't it the hardware stomach is what they call that? Where you would put, a magnet down there in case they got into something that they shouldn't, and then when it comes to the rumen, I heard it best described as just a big fermentation vat, right? And stuff sits in there until it, becomes small enough to flow. And that, I like that roundabout analogy because it is kind of like there's actually a little speed bump, right? That initial contraction to get back into that reticulum and then keep that flow going. I'm going to use that one. I'm steal that one.
So when we get to they're not called, we don't call cattle reticulites. They're not abomasimists or something. They're ruminants named after their rumen. You referenced that it's a kind of a big fermentation vat, Luke. What size in a mature dairy cow? I know steers are going to be a little bit different, but what size vat are we actually talking about?
Yeah, so I guess and Dr. Baker, you've probably done some cannulated animals experiments, but what I've found is I can fit the contents in a 40-gallon trash bag, 40-gallon trash can usually, but then it can. The way the metrics I've heard is it can hold anywhere between 40 to 60 gallons in a full-sized animal. So, it's quite the experience to pull that amount of material out, do the research and then put it back in.
Wow.
Pretty interesting.
So when it does, we'll get back to a little bit more about rumen in a minute, but to continue that highway analogy, we've come back through our traffic circle. What's going to go on? Where's that feed going next?
Okay. So from there, if the particle size is small enough, it'll flow to the omasum. And think of the omasum as kind of, it's a filter, right? It's made up of multiple layers of epithelial tissue. So then that way that digestive will flow through that, and that's what's screening out big particles and letting small particles through. But the key point of why it's multiple layers is absorption. So if you think about digesting the rumen, you're going to be, I don't know, what do you think, Dr. Luke? 3 or 4, maybe 5% dry matter. It's a lot of water.
And yeah, for sure.
And so you got to suck that water out, and that's what part of that omasum is. That's why it's so many layers, is to get that water out before it hits the abomasum, or what most people call the true stomach, right? So if we think about ourselves, our stomach, is full of stomach acid, right? And obviously there's some proteases and things that occur in there as well. But if you hit your acidic stomach full of all that water, and, Dr. Luke just said, there's anywhere from 40 to 60 gallons of water in there, theoretically. That's going to dilute your stomach acid down quite a bit. So you got to pull out that water before it hits that stomach acid, so you can actually have the acid do what it needs to do and further break down that feed stuff.
Perfect. So now we're going to go into our fourth compartment, our fourth stomach, what you reference as a true stomach. What's happening there?
Well, part of that acid, right? So, if you think, well, I probably not. If you want to get wild and think about the structure of proteins with how they're balled up in their quaternary or tertiary structures and all that jazz, those stomach acids at a low pH will actually straighten out those amino acids. So then that lets things like trypsin, which would be like a proteolytic enzyme in the stomach, come in and cleave off those amino acids. So then it'll be able to flow down in the small intestine for absorption. So that the acidity, there's a couple things that happen with acidity. Also part of it, like on our end, it helps kind of sterilize things. A lot of pathogenic bacteria, right, Dr. Luke, are killed by that true acidity. But the big part from a digestibility standpoint, you're going to see on that protein side, proteins are very, very susceptible to lower pHs, right? So they'll take away that structure of them.
Yeah, the abomasum, that true stomach, right, that resembles ours is a pretty important screening tool for both monogastrics and ruminants in that case, where you have nutrients that need to be denatured, like proteins, and you have foreign particles getting in there, foreign living agents getting in there that could go into that hindgut, establish infection. So that's just another hurdle, an innate hurdle that these microbes need to get through before they take over and cause problems.
Well, circling back then to the rumen, and I don't even remember where, who, what, but I've seen several presentations on this type of topic, and people seem to love to put up a picture of a bison or a white-tailed deer, something that is living on forage and nothing else type of thing, and say that in its purest form, this animal has a rumen that lets it eat these really low quality feeds, because then in her rumen these things can happen. We're working with the same layout, I understand that, but in either case, a high producing steer, dairy cow, or a bison, what's in the rumen that's breaking breaking all the feed down, but what's in the rumen that's letting her tuck into alfalfa and corn silage in a way that my family doesn't want to?
Well, it's the microbes, of course, right? They're the ones. They're the key players. And Dr. Baker, I don't know, I can kind of take this one a little bit.
You got it. You're the microbiologist.
Yeah, it's definitely something that separates that foregut, that fermentation vat. Those microbes that are housed there really separate the ability of these ruminants to degrade these celluloses from forages in a way that no other animals on the planet can. Yeah, I know termites. Yeah, but mammals, let's say that. No other mammals can, right? So let's go there. But it's super neat because, these are very specialized microbes that do this work. They require very specific conditions, and the cow, the ruminant animal, provides those things, that being a specific temperature. So that body temperature that's regulated by that host is maintained. The host provides food by consuming it and getting it to those microbes. The host keeps it an anaerobic environment, and provides enough water for all of that metabolism, all of that synthesis and things that happen there to take place. And it's really so I guess to kind of put it in perspective, we're looking at probably tens of thousands of different species, depending on what metrics you're using, whether that be using genomic technologies. Regardless, we've cultured, I think at this point, 500 to 1,000 different species, but we estimate many, many more. And they all come in varying abundances, so much so that they estimate an ounce of rumen fluid contains 1 trillion bacterial cells, or microbe cells, mostly bacterial. That's anywhere from 50 to 90% of the total biomass in the rumen. Let's just take 1 trillion cells per ounce, and there's, how many ounces per gallon. I think I did quick math on 40 gallons. I had to get my calculator out, take my shoes off, do some counting. But yeah, it's like 5,000 ounces in 40 gallons of rumen content. And so if you use that number, you can expect in the quadrillions in the rumen, total cells in there from all of these microbes. And it's staggering to think about. And another layer of that intricacy is that all of these microbes have to live together in this community and interact with one another and are facilitated by the host. So as much as you think cows are silly little animals, silly big animals that hang out in the pasture and walk around and mosey all day, they've got quite the system going on in their gut. And we're just scratching the surface on the research level, and so it's been really exciting to be a part of looking into that.
So it might not be completely accurate, but it would be, close enough. We used to say something was close enough for government work. I don't know if that's safe to say anymore.
Horseshoes and hand grenades.
There you are. That when a cow is eating forages, she actually can't get any benefit out of those. It's actually the rumen microbes that we're feeding, and then she's getting her nutrition off of those rumen microbes.
That's exactly right. And it's kind of like a weird analogy where we're, as farmers, farming and raising these livestock up, and then themselves are kind of have their own, herd of microbes in their gut that they're raising up and providing for. So in a way, they're farmers too, right?
So what you're telling me is even the biggest 10,000 cow dairy is an individual cow's keeping a lot more things alive than that guy with a 10,000 cow dairy.
Yeah, feeding a quadrillion little microbes probably cuts a big check.
We hear people talk about forage quality. I'll be honest. I think sometimes people want to ignore the wonder that is a ruminant and wish they were just monogastrics. And if we could feed them the way we do hogs or chickens, it would be so simple, and we're just going to pound grain. And I think that's a wasted opportunity. Why is an animal able to get more out of higher quality forages? If she's got what it takes to break down fiber, why do we spend any time talking about the benefit that can come from forage quality?
Well,
let Dr. Baker take this one.
Well, I mean, the big part of it is there's really nothing that can, well, I shouldn't say nothing. It is really, really challenging to digest lignin, right? And so as forages mature, they end up having more lignin. So I always kind of classically think of, like tree bark and things like that, right? It's full of lignin. So as forages, as they grow, they put up more lignin, that's just part of their life cycle. But yeah, ruminants really struggle to ferment those. To my knowledge, Dr. Luke could probably say, oh yeah, that sounds right. You need aerobic respiration to really break down lignin.
Right. Yeah. It's brown rot and white rot, right? So,
yeah.
Well, white rot actually technically, and
so,
mostly fungus, right? Mostly fungus, not even bacteria.
So obviously in an anaerobic environment, you can't have that. And so, no, I mean, when it comes to forage quality, ruminants can do quite a bit, right? But what always comes into play, on the dairy side, on the beef as well, is passage rate. And so, the longer it takes something to sit in the rumen, the less that animal's going to eat and the less that animal's going to produce. So, I mean, think of it as energy going in and energy going out. If you've got a delay in there, there's not as much energy going out, right? So, that's where, when it comes to a lot of like high efficient or high productive animals, yeah, you got to push a lot of good quality forages to keep that passage rate up, keep that digestion flowing, and keep that animal eating. Because if they're not eating, they're not gaining. If they're not gaining, or if they're not producing milk either, it's about kind of getting that throughput of feed intake. And so speed of digestion is definitely essential.
Yeah, and that to some degree is the same kind of ballpark idea of you'd call it, well, just improving your digestibility to get your feed to gain ratios. Like you want that feed to be broken down as much as possible, because I think that, you would agree that the biggest bill on the farm is feed costs, right?
Mm-hmm.
So maximize it and put those high quality forages in that are low lignin, that the microbes can break down and more rapidly, and get the maximum amount of energy out of that in the time that it's there.
I know, because he started a couple months before I did back in '93, that our former vice president of R&D, Ben Billy, one of the first things he did at Agri-King was to essentially create the only artificial rumens that have lived nonstop for the last 30 plus years now. What has that meant as far as our ability to better value feeds? How are we analyzing? What are the things that you guys can use that technology to get a jump? Obviously we don't do anything only in the artificial rumen, but as far as narrowing down the possibilities before we do take it to the field, how are those artificial rumens an asset to us?
Yeah, so I think, I like working with those things. I also like working with real living rumen content. There are some differences, absolutely. But the reality is, by bringing those, let's say, that outside microbial community inside, we are basically controlling the environment that they are living in. And that allows us to cut down on a lot of the variables or the variation that you would classically see in a herd of cattle. You don't have breed effects. You don't have environmental effects. You feed the same diet every day at the same time, kind of a thing. So you don't have these fluctuations or these random events that throw chaos into the mix. And by having that control, that structure, we can really have we have a really good way to compare feedstuffs and compare the digestibility of, what we call ex vivo or rumen microbes that were taken from the rumen originally and then transplanted into these systems that we control. So we know generally that chaos has a really big impact on digestibility, on the utilization of that feedstock. And by doing that, we can fit a digestibility number to an individual forage or individual nutrient or feed stuff. And that helps us build rations. Would you agree, Dr. Baker?
Yeah. You said it perfectly. I think variation. There's quite a few studies where even they took two steers side by side, emptied the rumen contents, and they measured microbial population, said, okay, well, steer one has 30% of this microbe family, right, or species, and then steer two has 40%, and they'll swap rumen contents. And I believe it's about it would be about a week, if I recall. You've read a lot of those studies too, I assume, Dr. Luke, before they return to their original population. And so, when you start trying to do like in situ work, which a lot of people do in situ, you can't use that same animal for year after year after year. And so when you start thinking like, well, 2 years ago we had, really great milk output, what is the difference between here and there? Well, then, so you've got potential variation in your NDF digestibility number that you're getting. along with, just obvious variation between year to year. And so by using those, we're taking out some of the variation.
Perfect. You guys have given me a lot to think about. I'm sure other people are sort of in that same, still trying to get their head around the quadrillion little things being taken care of in that cow's rumen. Any closing thoughts or observations? You guys obviously are smart. You could be doing anything for a living, and you choose to spend your professional life dealing with ruminants and rumens. Any final thoughts on that?
Well, I guess we could bring up the number that everybody talks about, just to put it in perspective of, the importance to animal production that ruminant in dairy animals, probably more specifically, but to some degree in beef. We can expect that the microbial activity in that rumen degrading that feed stuff and basically creating fermentation end products or waste products that then the animal itself can absorb and utilize as energy. That process accounts for, a lot of people say 70 to 80% of the entire energy requirements of that animal. So that, that to me says a lot, says that, that process of basically those microbes are mandatorily there. It's the way that these organisms will thrive. The host will thrive because the symbiosis between the microbial community that it's holding on to and the products that microbial community makes in turn. So it's really a big impact for these animals, and it's really, in my mind, I know people can say that they'll live without or defaunated rumens and they'll be fine, but really you're not going to get the maximum production. You're not going to get maximum health without these microbes. They're really important.
Perfect. Adrian, any closing thoughts?
Well, I would just keep in mind, if you look at, ruminants versus kind of some of the other animals involved in agriculture, there is a lot of cellulose that gets pumped out there, and the ruminants are, they're essentially the waste bin of human food production. You look at like things like high fructose corn syrup and corn gluten coming off that. Yeah, can you feed those to monogastrics? But will they fully be able to utilize all that fermentable fiber compared to a ruminant? And then if you just look at our total land mass, and even in the U.S., so much of that ground isn't tillable. A lot of it's in pasture. That's all it can be utilized for pasture. And so when you think about that cellulose mass that's coming off that pasture, it needs to be utilized. And ruminants are one of the only animals that can really utilize it.
Yeah, absolutely. I completely agree.
I read an observation over the weekend that said, even so congratulations, you decided to be a vegan for whatever strange reason, and you're now going to drink soy milk, but one of the only things that comes close to making that really work is that we can feed the soy hulls that used to wrap almond milk. We can feed that almond hull to a ruminant to get more use out of. And the same with distillers grains that come off of ethanol. And the amount, just what Adrian said, the amount of, co-feeds, byproduct feeds that we can then get more human nutrition out of by running them through ruminants would stagger people that have never given it any thought.
And that's another wild aspect, I think, too, and Adrian, you'll have to give me a number here. How many different feedstuffs do you think you've fed in your day? Just, it could be all kinds of things from almond hulls to gummy bears to and the rumen has to be able, to adapt to that. And that's where those quadrillion microbes come into play. That's where those tens of thousands of species have basically all the metabolic capabilities that you could ask for. in terms of harvesting energy. We did talk about lignin being the challenge there, but it is really staggering to think about how adaptable that rumen has to be to and it's not just the United States, it's all over the globe. I mean, what are those animals living on? What do they get day to day that they have to turn into, muscle mass or milk or something, pretty wild.
Well, guys, thank you very much. Chris, what did we learn today?
Hey, I am super fascinated with both these guys' passion. I've said this before. It's oddities, but the fact that one of you guys said that it's a little community that's inside these animals, and it's just walking around instead of just the cow that we see, and the tens of millions of whatever cells that are working all the time, complete with roundabouts. Didn't know that. So that's what I learned today. All right. Thank you so much for listening today. If you like what you heard, follow us on any of our socials. And if you have any questions or any comments that you'd like us to follow up on, you can hit us up at podcasts@agriking.com. All right. Thank you very much, Dr. Luke, Dr. Adrian, and Mike. Thank you so much and have a great day.
Thank you, doctors, and thank you, Chris, so much. Everyone have a great day.